Oh. Hello, everyone. Thanks for joining our session. It's a pleasure to introduce Dr. Sergi Regot from Johns Hopkins University as our next speaker. Sergi completed a Bachelor's of Science in Human Biology in 2006 and a PhD in Biomedicine in 2011 from Pompeu Fabra University under the guidance of Dr. Francesc Posas. After that, he was a postdoctoral fellow with Marcus Covert at Stanford University until 2015, when he joined Johns Hopkins University as a professor in the Department of Molecular Biology and Genetics. Sergi is the recipient of several academic awards, including most recently a Career Award from the National Science Foundation and a Research Scholar Award from the American Cancer Society. Sergi has been a pioneer in understanding how the dynamics of signaling networks impact the behavior of single cells, and his work has been both informative and inspirational for many scientists, including myself. I think I speak for everyone when I say we're really looking forward to your talk.
All right, so first of all, I'd like to thank the organizers for giving me the opportunity of presenting our work here today. My lab is interested in understanding how cell signaling in individual cells allows for coordination of collective cell behaviors in tissues. Over the last 50 years, and through a series of genetic and biochemical approaches, researchers have identified most of the components of mammalian signaling networks. However, if one tries to predict a cellular outcome based on the topology of one of these networks and Boolean logic, you would realize that it is still highly unreliable and very much cell type dependent. So in order to address this issue, we have to be a little bit more quantitative and identify those nodes of regulation that are more relevant in each case. But the problem with being more quantitative is that individual cells, even isogenic cells living in the same environment, can be extremely variable. And this is due to a variety of extrinsic and intrinsic factors, where extrinsic factors are other cellular processes that are not necessarily synchronized between cells in the population, and intrinsic factors have to do more with the stochastic nature of molecular interactions. Now, all these cell-to-cell variation make it such that bulk population assays are highly misleading. So the next question then is, how can we be more quantitative but have single cell readouts? And the way we like to approach these is by using signaling biosensors. These are genetically encoded probes, fluorescent probes, that allow you to measure cell signaling in individual cells by live imaging. And there's a different varieties of these kinds of biosensors. There's naturally occurring biosensors, like the ones I'm showing here. These are proteins that undergo a change in localization, for instance, upon a certain signaling stimulus, or a change in abundance, again, upon some signaling stimulus, but they are naturally occurring proteins. So if we just tag these proteins with a fluorescent probe, we can measure signaling over time in individual cells. Now, besides naturally occurring biosensors, there's also a variety of synthetic biosensors that allow you to measure things like metabolites, GTPase activity, or even kinase activity, as is the case of these kinase translocation reporters that work as follows. So basically, these are synthetically engineered substrates that have a fluorescent protein and about a 50 amino acid peptide that contains a docking site for a kinase of interest, an NLS, and an NES, in the context of two phosphorylation sites. So the way they work is when there is no kinase activity, these two sites are not phosphorylated and the NLS is stronger than the NES, so the fluorescent signal is mostly nuclear. But when the kinase is activated, it will bind a docking site, phosphorylate these two sites, inhibit the NLS, enhance the NES, and you will end up with a net translocation of the sensor from the nucleus to the cytoplasm that is very easy to measure by live cell microscopy. Now, the other interesting thing about these sensors is that they use one color per kinase, so we can use different fluorescent colors and multiplex more than one. As I'm showing here, this cell line has 4 different fluorescent proteins, one just dedicated to tag the nucleus for segmentation and analysis, and 3 other fluorescent proteins that allow you to measure ERK, JNK, and P38 activities in single cells over time. And these are not stills, these are videos, where just by calculating their nuclear to cytoplasmic ratio in each cell, we can measure the signaling activities in those cells over time. So the next question is, why do we want to use live cell imaging instead of using fixed cell approaches like immunofluorescence? And the answer to this question for me is twofold. First, because it enables measuring temporal correlations. That means if we had 2 biosensors in the same cell, we could know which one of the 2 pathways gets activated first with a relatively high temporal resolution. And in addition, and this is particularly important to us, it allows to compare the temporal dynamics of different cells within a tissue. But in addition, by doing these experiments, we have found, and others, that inferring dynamics from endpoint immunofluorescence analysis can be relatively misleading. And this is, for example, the case of NF-kappa B dynamics, where after stimulation, in this case is with TNF, the transcription factor oscillates in and out of the nucleus. And this kind of oscillatory dynamics has been observed also for other pathways, not just NF-kappa B, like P53, ERK, and others. And at the end, what that means is that if we try to infer dynamics by looking at endpoint immunofluorescence, it can also be relatively misleading. So now I'd like to share with you 3 different stories from the lab where we leverage live cell imaging of signaling biosensors to address collective cell behavior in tissues. First, I'm going to talk about work by Team Aking in the lab, who had the following question: how oncogene expression in tissues alters signaling dynamics in the cells that express the oncogene, but also in the neighboring cells. And in addition, how signaling dynamics can inform cancer phenotypes. So the way he went about this was he created a cell line based on the human breast mammary epithelial cell line MCF10A that is non-transformed and chromosomally normal, where he could measure ERK activity by expression of the ERK KTR. As you can see, this is a very dynamic biosensor that again shows these oscillatory pulsatile dynamics under basal conditions. Now, in addition to the ERK KTR, Tim introduced in this cell line a TED-on system where he could induce the expression of an oncogene and measure dynamics in real time. Then he tried about 15 to 20 different oncogenes, and as a result, he found that all oncogenes can be classified in 2 different classes. One class that will increase the pulsatile dynamics, meaning increase the frequency of the ERK activity pulses in those cells, and another set of oncogenes that increase the activity once and only once, and it remains sustained over time. So we call these sustained oncogenes and pulsatile oncogenes to differentiate them. Now, in terms of function, what he did was compare some of the oncogenes that created pulsatile ERK activity and some that created sustained ERK activity and measured different phenotypes. This was measured by EDU incorporation, so what's the fraction of cell cycling cells in the population? And he could see that all the oncogenes that increase pulsatile ERK activity have also increased cell cycle proliferation. However, if you look at the genes that have sustained ERK activity, they have a very strong cell cycle arrest. We're now trying to investigate the mechanism for these arrests, but it really resembles the previously reported oncogene-induced senescence. Now, we totally believe that these is dynamics dependent and only dynamics dependent for 2 reasons. First, if you compare BRAF wild type and V600E, these 2 kinases are exactly the same. They have the same substrates, but they differ by 1 point mutations that renders the V600E mutant hyperactive or constitutively active. but also because if you induce these kind of dynamics at different levels, as it's done here with EGFR or MEK2, you can trigger the same phenotypic changes: increased proliferation when its ERK activity is pulsatile, and decreased proliferation when ERK activity is sustained. And in addition to these opposed dynamics-dependent cell fates on cell cycle, we can also see a change in cell migration. But the point of this study was not only to look at cell-autonomous phenotypes, but look at what happens to the neighboring cells. So what Tim did was now label the oncogene expressing cells with a different fluorescent protein, a green fluorescent protein, and induced with doxycycline or not, and measured the activity in the cells that express the oncogenes, shown in green up here, but also the activity on the neighboring cells that do not express the oncogene. And strikingly, what he saw was that certain oncogenes, which again were only those that trigger sustained ERK activity, not the ones that trigger pulsatile ERK activity, were capable of triggering waves of ERK activity through the neighboring cells. And I'm going to show you here an example of this striking finding, where there's 3 examples, and you can see the green cells activate, but then the neighboring cells have these waves of ERK activity. I'm going to pause this and play it again. These cells activate first, and then the neighboring cells experience a wave of ERK activity. So that was interesting to us, and we tried to identify mechanistically what is behind these waves. We found out 2 things. First, that they depend on EGF receptor signaling. If one adds EGF receptor inhibitor, you can block the neighboring cell activity while not affecting the oncogenic induction of ERK activity. But also, we found that it depends on the matrix metalloprotease ADAM17, which usually sits in the membrane and is capable of shedding factors such as EGF-like molecules. So what Tim did was make a knockout of ADAM17, this protease, and use those cells as the oncogene expressing cells here, or the neighboring cells. And what he found is that if the neighboring cells are ADAM17, there is no difference. Cells can still experience these waves of ERK activity. But if the oncogenic cells are knocked out for the protease ADAM17, now the neighboring cells are not experiencing ERK signaling waves. So in conclusion, what we think is happening is that sustained ERK activity is somehow decoded by the matrix metalloprotease ADAM17. And once it's activated, it can cleave growth factor that then diffuses to the neighboring cells and activates these waves of ERK signaling. But what is the function of lateral signaling? So since these are epithelial cells and what we see is EGF receptor signaling, one possibility is that it triggers proliferation of neighboring cells. And that's exactly what we see. So even though the oncogene expressing cells are arrested and suffer this oncogene induced senescence-like phenotype where upon sustained ERK activity, they reduce the cell cycle fraction. The neighboring cells, however, this is a co-culture with only 10% of RAF V600E cells. The neighboring cells increase proliferation by more than tenfold, by around tenfold. So this is very interesting to us because it means that signaling dynamics, depending on the oncogene that those cells accumulate, can dictate non-cell autonomous contributions to tissue growth. And it's actually the wild-type cells that are growing around it. But Tim also realized something else, and when he was looking at these movies, he realized that the neighboring cells have a very striking directed migration upon induction of the oncogene towards the green cells. And I'm going to show this in this video where we induce with doxycycline and the neighboring cells tend to all clump around the green cells. I'm just going to go back and forth in this video. And as you can see, there is a very striking directed migration towards the green cells that he could quantify by measuring the angle of motion of each cell towards the nearest green cell. Upon addition of doxycycline, there is a strong bias, and this bias depends again on EGF receptor signaling and ADAM17. So in looking through the literature and thinking about it, we thought that maybe this directed migration of neighboring cells was contributing to the so-called oncogenic cell extrusion, by which cells are pushed, oncogenic cells are pushed out of the monolayer. So to address this question, what he did was using a confocal and plotting Z projections with a Z stack. He could see that upon addition of doxycycline, the green cells are clearly up from the monolayer, so they are being extruded. We came up with a way of quantifying this extrusion. Again, we could see that extrusion is dynamics dependent. Only oncogenes that have sustained ERK activity are capable of extruding. And also, he found that this extrusion depends on the matrix metalloprotease ADAM17. So if there is no lateral signaling, extrusion does not happen. So what we believe is happening is that depending on whether the oncogene triggers pulsatile or sustained ERK signaling, we will have two very different scenarios. One, in the case of pulsatile ERK signaling, you will have cell autonomous proliferation. And in the case of sustained ERK signaling, you will have a cell cycle arrest. But then due to activation of ADAM17 and shedding of EGF receptor ligands, in this case amphiregulin, the neighboring cells will now be the ones proliferating, and the oncogenic cells will be extruded from the monolayer. Now, whether this extrusion is protective from oncogenic process and development of malignant tumors, or it contributes to metastasis, we don't know yet. So now I'm going to change gears a little bit, and I'm going to tell you about some of Helen's work. So Helen is a graduate student in the lab that had the following questions. How do epithelial tissues quantify pathogen-associated molecular patterns? So what she did was similar to what Tim did, using live cell biosensors to measure NF-kappa B signaling dynamics in epithelial tissues, again, breast mammary epithelial cells grown in a monolayer, and stimulated those cells with a large variety of pathogen-associated molecular patterns and cytokines that are known to trigger NF-kappa B activity, and measured both the translocation, the nuclear translocation of NF-kappa B. This is heat maps where each line is a single cell, and each row is time, and we have the localization and the gene expression in tandem for each cell. So this summarizes about 18,000 cells in one experiment. And what she found from this screen was that the bacterial lipopeptide PAM3CSK4 triggers a very all or none response in epithelial tissues. She verified these using primary organoids and breast organoids and gut organoids. Now, this bimodal behavior was very striking to us. And so what she did was try to stimulate cells with other molecules that are not necessarily in their initial screen, but that signal also through either TLR1 or 2. And what she found is that all the molecules she tried that signal through TLR2 or a dimer of TLR2 triggered this digital behavior. And interestingly, even though she increased the dose by 1,000-fold, the response seems to be capped at around 12–13% of cells. This is variable between experiments somewhat, but it's pretty hard set at about between 10 to 15% of cells. So it seems like the upper limit of the amount of responders is somewhat limited at the population level. Now, since this was all TLR2 dependent inputs, what Helen found out is that the expression of TLR2 was limiting. So she induced heterologous expression of TLR2 and could see that now all cells become responders to these lipopeptides. And in addition, she used single molecule mRNA FISH and found that the responder cells are significantly expressing more TLR2. In fact, the non-responder cells are almost non-expressors. So how is this TLR2 expression being regulated was the next question. And what Helen found out through the literature is that there are several papers that link epigenetic regulation to TLR2 expression. So she tried a few epigenetic regulators and found that when we inhibit DNA methylase activity in cells, we can increase the fraction of responders dramatically to about 60–70%. So we thought that maybe the expression of TLR2 was controlled by methylation. So in order to measure methylation in single cells, we couldn't necessarily do that, but what she did was use nanopore to do it at the single molecule level. So when we do nanopore sequencing and we can get methylation reads, we can see how the methylation of the TLR2 promoter is bimodal as well. So methylation occurs in block, so either all methylated or all unmethylated, and the fraction of unmethylated versus methylated reads maps very well the fraction of responders and non-responders in her functional assays. So then we did some modeling and we realized that there is a certain balance here where cells have a probability of switching from responder to non-responder and from non-responder to responder. But more importantly, Helen showed that if you incubate these tissues with other inflammatory molecules, these balances of switching from responder to non-responder or the other way around can be modulated. And when that happens, the fraction of responders at the tissue level changes. So what we believe is happening is that the bimodal expression of TLR2 allows for a tissue level response. So at the tissue level, there is an epigenetically regulated digital signaling that defines innate immunity. Now, again, I'm going to switch gears and talk to you about my last example, which is Mike's work. Mike is also a grad student in the lab, and he had the following question: how MAP kinase signaling dynamics patterns the mammalian pre-implantation embryo. Now, in mouse pre-implantation development, what happens is the first few divisions are generating the same type of cells, and you end up with a morula that has about 16 to 32 cells, which eventually develops a cavity inside, separating what we call the trophoectoderm and the inner cell mass. Now, in the next step, this inner cell mass further segregates into lineages, one called the epiblast, that it's marked by nanog expression, and one that is called the primitive endoderm that's marked by GATA6 expression. Now, through the literature, it is clear that ERK activity is involved in this segregation, because if you add FGF4, all cells choose the primitive endoderm phenotype. But if you add an ERK inhibitor or do an FGF4 knockout, all the cells choose the epiblast fate. So what Mike set out to do is, can we actually express the ERK KTR in mouse and measure ERK signaling dynamics while this specification is taking place? So he first developed the mice, and the first thing that he realized is that most of the cells of the inner cell mass are actually active on ERK activity. But based on the genetic evidence, we would expect at least a fraction of cells that are low on ERK activity. So that was surprising. But again, the beauty of this approach is to do live cell imaging. So he used confocal, spinning disc confocal, to acquire Z-stacks over time, and he made the following discovery. I'm going to play a video here. This is the H2B signal. This is the ERK KTR signal. And I'm going to point out at a cell that has a very brief pulse of ERK inactivity. So while most cells are active all the time, you'll see here, there, how at some point, for a brief period of about 2 hours, some cells experience these dips of ERK inactivity that were the only time in this inner cell mass development where we could see significantly low ERK activity. So then he measured individual traces of a few embryos, and the striking finding was that this inhibition of ERK activity only occurs after mitosis. At no other point during the cell cycle there is such an ERK low activity. Again, these are heat maps where each row is a single cell and each column is a time. The blue marks mitotic events, and after every blue you have the two daughter cells. And for example, this is an ERK, what we call an ERK low division, and these would be an ERK high division where cells resume with high ERK activity after division. Now, to make it more clear, we've now done an in silico synchronization of many of these events, and we can confirm that it's right after mitosis that cells experience an ERK low activity or ERK high. So we think there is a bifurcation of ERK activity after mitosis that could explain the segregation of primitive endoderm and epiblast in the mammalian preimplantation embryo. But to prove that, he had to go one step further and identify which is the fate of each one of these cells. So what he did was doing the live imaging experiment, then fix the embryos, use antibodies to do an immunostain, and then re-image those embryos to register and identify which cells are which from the live imaging to the fixed experiment, and then bin all those traces, the ones that are ERK low divisions and the ones that are ERK high divisions, and quantify their levels of NANOG and GATA6 after the imaging experiment. And what he found was that, in fact, if you plot the levels of NANOG and GATA6 in each individual cell and color the dots depending on the ERK activity right after mitosis, there is a striking correlation where the cells that have ERK low activity after mitosis tend to have higher NANOG expression, but the cells that have ERK high after mitosis tend to have GATA6 expression. But if you correlate the activity with the last time point, just as a control, there is no such correlation. So we believe that it is the activity of ERK after mitosis that helps tilt the balance of the NANOG GATA6 expression level and favor for the ERK low cells NANOG activity and therefore epiblast fate. Again, this would be the summary where cells can undergo one of two different fates after mitosis. We have the ERK low divisions and the ERK high divisions. And those cells that experience an ERK low division after mitosis, they will become epiblast, they will increase NANOG expression, while the ERK high cells become primitive endoderm. And so as a conclusion, it looks like mitosis desensitizes some cells stochastically to FGF signaling, which is present all throughout this embryo, to push the NANOG GATA6 equilibrium towards NANOG. So as a summary of everything, we believe that the main advantages of studying signaling with live cell biosensors are that they capture very heterogeneous behaviors, that you can get very high temporal resolution, and also differentiate cell autonomous versus non-cell autonomous behaviors. And with this, I want to thank the whole lab. I've talked about Mike's team and Helen's data, also our collaborators and our funding sources. And I will take any question.
Great. Okay, so we have one question. what's the mechanism underlying ERK activity bifurcation upon mitotic exit in mammalian embryos?
Yeah, so this has been part of our focus lately. We were lucky enough that in embryonic stem cells, this bifurcation also occurs. So there we can do a lot more editing and perturbations than in the embryos. And it turns out that what we think is happening is that during mitosis, the anaphase promoting complex, the APC complex, degrades the MAP3 kinase, RAF, that is responsible for transducing the signal from FGF down to ERK. So that happens stochastically for different periods of time, and in those cells where it happens completely, they become desensitized, and then they cannot transmit the ERK signal from the receptor to the kinase, and therefore its activity is low. Nano gets stabilized, and the balance between nanog and GATA6 gets shifted towards epiblast.
Awesome. Any more questions? Well, I guess not. So I guess we can close the Q&A and thank the speaker for an amazing talk.
Thank you, David.